So welcome to Sano Seminar. Today we host Professor Marek Pruszyński from Novaten Center. Novaten Center is located at Świerk in at the Institute of Nuclear Research in Poland. And Professor Marek Pruszyński is obtained his PhD and habilitation in both in chemistry, and he's expert in using radiopharmaceuticals for different purposes. And our seminar today has a hybrid form. So we have participants at Sano, together with Professor Pruszyński, and we have also participants online. So, as usually, we will, if you have an urgent question, really required to understand the talk, please ask it on questions and answers or on chat. I will try to transfer this question. And as usual, we will have a discussion after the talk. So, Marek, floor and screen also is yours.
Okay, thank you very much for the nice introduction, and thank you also for inviting me here. I really wanted to come in person because I also wanted to get to know you. I know that you are working on the computational studies and work. It's somehow totally different from this what we are doing at our group. We are very experimental, but I think that at some moment we need both. We need experiments and data for the modeling and computation, and vice versa. We also need computation. For example, if we synthesize some compounds, it takes a lot of time. So to speed it up, maybe the computation modeling will support us. So maybe we can find something joined together. The title is related to novel radiopharmaceuticals, from the production, from the radionuclide to the clinical translation. It's a really long process, and I will try to explain to you how does it work. So what are the radiopharmaceuticals? As by name, it's a combination of two major components, the radionuclides and the pharmaceuticals. And the pharmaceutical might be even a simple chemical compound or a biomolecule, which is also, for us as a chemist, for me, it's also a chemical compound, but much more complicated than a simple one. What are the properties of those radiopharmaceuticals? Definitely, they have to accumulate in special tissues or organs, because we want to use those radiopharmaceuticals for diagnosis, for diagnostic or therapeutic purposes. And in case of radiopharmaceuticals, we can divide them for several classes. There are several classifications. One of them is based on the structure. We have simple radiopharmaceuticals, like simple inorganic salts, sodium iodide, radium fluoride. We have small molecules, like simple organic compounds. There are like tens, maybe 100 atoms. That's it. But we have more complicated radiopharmaceuticals based on biomolecules, intact monoclonal antibodies, their fragments or peptides. To couple the radioisotope, we have to use a special linker. Either it's a prosthetic group or it's a chelator for metallic radionuclides. And in case of those radiopharmaceuticals, we can say about personalized medicine, because they are molecularly targeted radiopharmaceuticals. It means that the biomolecule interacts with the receptor, which is existing only on the cancer cell, or it's overexpressed over there. And the overexpression can be several magnitudes of order. What happens with the radiopharmaceutical whenever it's injected to the bloodstream, into the vein? So it can enter to the cancer cell just passively through the membrane or through some special transporters. For example, sodium iodide transporter is responsible for the transportation of iodine inside, but they can also interact with the cancer cell specifically. Here we have the receptor, the biomolecule with the radioisotope binds to this receptor, and it has three options. If the binding is weak, so it's dissociated and it's released. It can bind very strongly and stay on the receptor, or whenever it binds, it can also trigger a signal. So the entire complex is internalized inside. And in case of some radiopharmaceuticals, it's very important because if these are the short range, if the radionuclide emits particles like Auger electrons or alpha particles, it's very important to enter inside because the range of the emitted particles is like nanometers or micrometers. So to think about radiopharmaceuticals, first we need to think about radioisotopes, radionuclides. We need to produce them. They can, of course, they are existing naturally, so we can dig in the mines and try to extract. But the most common way to get the radioisotopes for the medical application is nuclear reactor or cyclotron. Here we have, for example, iodine-131. It's the most commonly used radioisotope in the world for the thyroid, for example, thyroid disease. And it's produced as a product of fission of uranium-235 in the reactor. Cyclotron, it's accelerate protons, deuterons, or alpha particle beams, bombard the target material, and the new radioisotope is produced. The third way is radionuclide generator. The mother radionuclide, which is produced in the reactor or cyclotron, is put on the special column. This radionuclide is absorbed on the resin, and the mother radionuclide decays to another radionuclide. It's another element, so in this situation, it just can be simply eluted from the column and used either directly for the intravenous injection or for the production of radiopharmaceuticals. And Nomaten Center of Excellence is located at National Center for Nuclear Research, which is around 30 to 40 kilometers from Warsaw. And over there, we have a very unique infrastructure for the radiopharmaceuticals development. First of all, everyone knows the Maria reactor. It's the only, so far, nuclear reactor in Poland, and it's a research one. It's not used for the energy production. It's used mostly for testing materials. There are many other groups at Nomaten for the material testing, heavy entropy alloys, for example. But also Maria produced many radioisotopes for medical application. 30% of worldwide iodine-31 is produced by Maria reactor. Here, on this picture, it's not visible, but on this picture, it's currently a new building which consists of a cyclotron. So another way of production of radioisotopes. So we have two sources currently. So that's why we are clearly independent from external producers. Between those two buildings is Polatam, which is the radioisotope center. They produce for example, from target material, they extract the radioisotopes and they sell it either to companies, hospitals, or they produce radiopharmaceuticals and distribute worldwide. And Nomaten is here. It's on the other corner. We have over there our offices. We have also our laboratories, but we also can use laboratories of those all units at NCBJ. So over there we have our radiochemical, biological laboratories, and also preclinical research where we have the SPECT-CT and PET-CT cameras for animals imaging. The other, how we can divide radiopharmaceuticals in other way, by the emitted particles. If they emit gamma radiation or positrons, we use them for diagnostic purposes. Why? Because gamma is very, how to say that? It goes out of our body. So that's why it can be detected by the gamma camera. For therapeutic purposes, we use beta minus particles, which are electrons. Radiochemists use Greek letters to differentiate where is the source of this electron. It's not from outer shell of the atom, but it's from the nuclei. The same alpha particle, it's a helium without electrons, but it's from the nuclei emitted, so that's why we call it alpha. They have a short range, that's why they use for the therapeutic application. They have a high energy, they destroy cancer cells, but they cannot go out of the body. And for diagnostic purposes, there are two major techniques. Single photon emission computed tomography, which is called SPEC. The isotope which is used over there emits gamma radiation, gamma line. And this gamma radiation has a specific energy, which might be detected by gamma camera. There are two planar gamma cameras, here and here, which circulate around the human body and collects this gamma quantum. And by this, it obtains several or hundreds of 2D images, which later are reconstructed into 3D. Over there is also CT, which is computed tomography. So also it have an image of morphological of the organs. Because in case of radiopharmaceuticals, we use mostly the image of accumulation. So we see what's going on with the radiopharmaceutical. This is the very important, that it interacts with the cells. In case of CT, we only see the image of tissues, organs. We don't see the processes ongoing. And in case of PET, which is positron emission tomography, here we use the radionuclides, which emits positrons, and positron is the anti-electron. So whenever it's released from the radioisotope, it immediately interacts with the electrons around, annihilation, and we have two quantum of gammas, very characteristic. 511 kiloelectronvolts energy, and of course 180 degrees emission. So here we already have like a device. Here we had two planar gamma cameras. Here we have crystals which collect 511 quantum gammas in circular way. So we already obtained the 3D image. Also from the CT, which is behind the PET, there might be also computed tomography, CT, or might be MRI, which is the magnetic resonance imaging. Currently, it's the MRI substitutes the CT, especially in case of brain tumors, because the image of the morphological tissues it's much better resolution. The most commonly used radiopharmaceutical currently in the world is FDG. It's a simple sugar, glucose, which in this position has this radioisotope fluorine-18, which decays of emission of positron with electron to quantum gammas, so we can do the imaging. Why glucose? Because it's the major source of energy for cells. And in case of tumor cells, they proliferate, they divide so quickly, they need a lot of energy. So they use a lot of sugar. However, this radiopharmaceutical is not fully specific. Why? For example, here, all those dots are, of course, metastasis. However, this one is not, it's a bladder. FDG is a small molecule, so it's filtrated by kidneys and it's released with the urine. So this spot is not a cancer here. Also not. Patient a few days before collapse, maybe not break a knee, but there was an inflammation. And again, if it's inflammation, repair of cells. So at that moment, also a high demand for the glucose. So to differentiate, a very good radiologist is required to differentiate if it's really a cancer cell or not. But we have also specific radiopharmaceuticals. For example, PSMA. PSMA is prostate-specific membrane antigen. It's a very simple molecule, 2 amino acids, lysine and the glutamate. Here are some small organic parts which influence on the lipophilicity of the molecule. So somehow it may interact better with the membranes or cross the brain-blood barrier. And here's the chelator, which complex the technetium-99 radioisotope. And if we look at the image from this SPECT and the image distribution of this radiopharmaceutical, again, we see kidney, small molecule, elimination through the kidney and the urine. But here is the metastasis of prostate cancer. What else? This specific amino acid peptide has also a tendency to accumulate in the salivary glands. We don't know why there is this overexpression of PSMA receptor in this part of the body, but it doesn't mean that there are cancer cells. In case of therapeutic radiopharmaceuticals, a very simple one used maybe like for already 8 years, 90, it's sodium iodide. Iodine is very important element for human body because in the thyroid it's transformed into hormones, tyrosine, which influence on our body and several processes that are ongoing. So iodine, after let's say swallowing of the gel tablet, goes to the stomach. Gelatin is destroyed, and the iodine is going to the bloodstream and goes to the thyroid and accumulates over there. That's why when we had this Chernobyl or Fukushima, let's say, accident, at that moment, to block the thyroid from the iodine, many people got either sodium iodide tablets or Lugola liquid to block the thyroid from the accumulation of radioisotope. However, when the iodine is accumulating over there, it decays emission of beta minus particles, which can destroy the cancer cell. Iodine is also used for the disease when the thyroid is growing too much. One of the options is just the resection, surgical, or to avoid this is giving the sodium iodide, and some of the thyroid cells are dying, and that's why it stops subnormal growing of this organ. The other one, very simple radiopharmaceutical, is again radium fluoride. Radium is in the same column like calcium, so it has exactly the same properties. Calcium accumulates in the bones, so radium also is going over there. So whenever there are the changes in the bones, they need new hydroxyapatite, they need calcium, so that's why also radium accumulates over there. Radium is 2 to 3 is used for the treatment of metastasis from the prostate cancer, because the metastases are mostly going into bones. Again, PSMA, here with the lutetium. Previously, there was technetium-99 used for diagnosis, but here lutetium emits beta minus particles, so that's why it's used for the therapy. Again, on the cancer cells, we have those receptors. PSMA drug is binding over there, release beta minus particles, and destroy the cancer cells. For example, here we see the patient with metastasis through the whole body, mostly in the bones. And for example, here the patient who was relapsing after the treatment with lutetium. So there was a good recovery, but at some moment the cancer stopped to respond. So doctors changed to actinium-225, which is the alpha particle emitter, short range. And after 4 doses, It was almost a full treatment. And this patient was treated in Warsaw, in Banacha Hospital, and the survival was more than 5 years. So it was a good, how to say that, good treatment and good prognosis, because I think this patient was lived even 10 years. So whenever we think about the radiopharmaceuticals and their development, there are many challenges. First of all, we have to decide what we want to treat, if we want to treat or do the diagnosis. Based on this, we can decide on the type of radioisotope. If we decide the radionuclide, it's very important, the half-life. As you see, for the diagnosis, most of them have short half-life, which means every 20 minutes, half of the radioisotope decays. It's very important for the diagnosis because we don't want to inject the patient with radioactivity and release after 1 hour of diagnosis to home, because this patient will irradiate others at that moment. But for therapy, it's good to use longer-lived isotopes because this radioactivity has to interact with the cancer cell for some time. If we know what we want to do and what kind of radionuclide we need to choose, we need to think about what kind of cancer we want to focus on. Based on that, we can decide if we want to have small molecules, organic or intact antibodies, or maybe some of their fragments. Later, how to synthesize them. And if we have a product, how to do the preclinical studies on cancer cells and on mice, if it's working or not. If the results are promising, we need to upscale. Before we proceed to the clinical trials, we need to have a lot of radioactivity, a lot of radiopharmaceutical. So that's why we need also automation processes. And after that, there are clinical trials and all the formalities, which takes also time, because at some moment, even if the radiopharmaceutical is ready, it's proven that it works. It sometimes takes even 3 to 4 years to get the approval from FDA or EMA. And what kind of carrier we would like to use for those radiopharmaceuticals? There are many of them. Small molecules, their advantages, very simple to synthesize. We don't need a lot of chemistry. We just choose few blocks, synthesize, introduce the radioisotope, and that's it. A small mass, fast pharmacokinetic. After injection, very quick delivery to the targeted tissue. Small molecules, they can be transferred between blood and the brain. So for glioma cancers, it's very good to use them. However, they are not specific as FDG. Peptides, it's another carrier. High activity, biological activity. So interaction with receptors on cancer cells, they can be easily synthesized because we have automatic peptide synthesizers. And still, again, the small molecule. So again, advantage as a small, fast pharmacokinetic. Monoclonal antibodies, they're much more complicated. Dozens of thousands of atoms. So first, someone has to discover this molecule. Later to produce in a GMP level, great. Later we can come and try to attach a radionuclide. But to attach, we need to develop a chemistry. So here it's much more complicated. However, the advantage is a very good interaction with the cancer cells. Slow pharmacokinetic, which I will show you also later on. Nanoparticles. Nanoparticles might be inorganic, might be organic, like micelles or lipids. They can be used for multimodality. Antibodies can be attached to the nanoparticles, so they can interact with the cancer cells. we can put their fluorescent dyes or luminescence dyes inside, so they can be also detected not only by, let's say, infrared or some special lasers and emission of luminescence light, so we can detect cancer cells in several ways. Also by radioactivity, we can incorporate radionuclides into the nanoparticles, which I will show also later on. So synthesis, I will not go into details. Sometimes it's many steps, but sometimes it's only 2 or 3 to get the final product. But it requires development. Later, when we have the product, we need to check how it interacts. If it's specific, for example, over there, we have SCOF3 cells, it's an ovarian cancer, and a nanobody was compared with the trastuzumab. Trastuzumab is a monoclonal antibody that interacts with the HER2 receptor. So, for example, we see that this nanobody was interacting with the HER2 on SCOF3. Trastuzumab was not competing, which is good, because trastuzumab is used currently as a first treatment in those kind of cancers. When we add too much excess of nanobody, binding to cancer cells was blocked, which means that it's indeed specific interaction. On the other cell line, there is no receptor, so no binding. So it's proven that it interacts with those cancer cells. Later, we check the binding affinity, cellular uptake, toxicity, and many other factors. The next step in the preclinical evaluation is, of course, in vivo imaging. So in mice or rats, because we can do this in our laboratories, we inject those radiopharmaceuticals. We do the imaging, so we can see that this was a nanobody. So that's why it's a small molecule. So it accumulates in the kidneys, but also in the tumor, which was inoculated here under the skin. But after, we can do the also ex vivo studies, and we can check in each organ how much activity was there. So, for example, we see that it's going steadily accumulation in the tumor. Later, it's slightly going down, but also kidney accumulation was visible. But the background is very low. And when we have the final product, before we proceed further, we need to do automatization. Why? We need to produce large quantities, big amounts radioactivity. So we cannot be exposed too much on that. So everything has to be done by automatic synthesizers, which have many valves, many pumps, many heating chambers, also HPLC columns for the purification. And everything is done in the hot cells. Hot cells, which means that they have lead filling like 20 centimeters to protect us from the internal radiation. So everything is done whenever the product is produced at high scale, it's done in the clean rooms. As you see, people are wearing special clothes which cover their head, nose and mouth, but also shoes. It's forbidden to wear any other stuff, even earrings, rings, because this might be a source of contamination. Women also cannot use the spray for hairs. I don't know how is it called in English. Lacquer. Hairspray, yeah, sorry. Because it might be also source of organic compounds contamination. Makeup is forbidden. After, yes, but not during entering to the special clean room. Why it's yellow here? It's not because of the light over there. Otherwise, the light was normal, white. But it was done through the window, which is with the LED. So glass containing LED has this yellowish color. If more lead is incorporated in the glass, it becomes more greenish even. And in most cases, the production is overnight. So yes, because the production overnight, at 5, it's divided into doses, packed to the boxes, given to the car, and car is shipping and distribute in the city or even outside, because from the Warsaw, they're also going to Gdańsk, to Krakow. Whenever the cyclotron, let's say in Warsaw, breaks, so Krakow company produces it here and also distributes to Warsaw. Very important in case of production of radiopharmaceuticals is also quality control. First of all, appearance, the color of the radiopharmaceutical. Mostly they're solutions, so they should be transparent. If they have different color, like yellow, so it means something gone wrong. It cannot go to the patient. Purity, purity in all factors. There cannot be any particulates inside. Why? Because if they are injected into the blood, they can clog the micro vessels, blood vessels inside of the body. Chemical identity. It has to be done also immediately after production, either by TLC or HPLC methods. pH. We cannot inject acidic compound, because otherwise the blood will coagulate and we will be in troubles. So all those factors are checked before it's shipped to the hospital. The other are done later on, because bacteriological or sterility tests, they need time. So that's why development of radiopharmaceutical takes so much time. It's the same like for normal pharmaceuticals. We can, let's say, we can visualize hundreds or thousands of compounds, but due to the synthesis and all the checkings, only one can enter to the production and can be used in nuclear medicine. And from, for example, my personal experience, at the beginning, I worked with the monoclonal antibodies. And here was the trastuzumab. Monoclonal antibody is built up from the heavy chain and the light chain. The molecular weight is 150 kilodaltons, which means 150,000 grams per mole. It's a huge molecule. So whenever we labeled this with the lutetium through the DTPA chelator and injected this to the mice, we realized that, okay, accumulation in tumor is growing with time. But after 3 days, we have the maximum accumulation. The pharmacokinetic is really too slow. For a long time, a lot of activity is in the blood. So it circulates, it irradiates the other organs. So it may cause toxicity. So we decided to focus on smaller parts of antibodies. At the beginning, we worked with the FABs, but later we entered the collaboration with Brussels, and we got from them nanobody. And nanobody is a part of the heavy chain antibodies. Those antibodies were by accident discovered in the camel's blood. And in this small fragment, all the CDRs, which are complementary determining regions, are on this one small part, and they interact specifically with the receptors. And we do the same. This HER2 antibody, we label it in the same chemistry. And if we can see, there is no background almost. Only in 2 organs we observed accumulation, tumor and kidneys. And kidneys, because the mass is 15 kilodaltons, so that's why. To reduce the accumulation in kidneys, we use several other options, which I will see in the meantime. But also we can use blocking agents for kidneys, and at that moment, the filtration is stopped and it's going directly to the urine. What we decided to use for iodines? For iodine, how to say that, coupling to the nanobody. The simple way for iodine introduction is just electrophilic substitution in the benzene ring, and the benzene ring is in the tyrosine. one of the amino acids which is commonly available in every molecule. The other one was synthesis of a special group, SGMIB, in which we introduced the iodine there. And the MALGIC was a third prosthetic group. From the experience on the monoclonal antibodies, MALGIC gave the promising results. And it was the best for the trastuzumab. However, when I switched to nanobody, I figured out that it's totally opposite. First of all, why normally we cannot use the electrophilic substitution for the tyrosine, because this is unstable in vivo. There is the enzyme, the iodinase, which recognizes that the iodine is next to the hydroxyl group in the benzene ring, and just cut it. Whenever it's cut, it's released to the blood. We observe the thyroid over time accumulation. For the MALGIC and SGMIB nanobody, there is no accumulation in the thyroid, which means they are stable in vivo. When we compare the tumor accumulation, MALGIC gave better results than the simple radioiodination. However, SGMIB was much better. At 24 hours, it was double accumulation. However, but the biggest difference was in kidneys. Simple iodination, so it means like a generally not changed nanobody. Accumulation in kidneys was going down with time. For SGMIB nanobody, it was tripled at the beginning, but later it was going down even much less than simple radioiodination, which was really surprised for us. It means that the metabolism in the kidney is totally different for this group, which speeds up even the release of the metabolized form. However, MALGIC was the worst. It was so much accumulating in the kidney that by treating the patient, let's say in the future with this radiopharmaceuticals, maybe we could treat the cancer, but cause the nephrotoxicity, which means that patient would need the dialysis up to the end of the life. Later on, I switched from the same nanobody, but to the actinium-225, which is the alpha emitter. And we decided to treat mice with simple saline and the nanobody, which was control. Control, it means it didn't react, it didn't bind with the HER2 receptor. And what we did before, in mice, we just intraperitoneally injected SCOF3 cells overexpressing HER2 receptor. And why we did it intraperitoneally? Like to mimic the spread of ovarian cancer. And we see that for the control groups, those cells were growing. And mice, at 52 days, we had to euthanize mice because it was already too much. And from the regulations, we had to sacrifice them. But one dose of our radiopharmaceutical already increased the survival twice. 3 doses, which were injected over like 3 days from the inoculation of cancer cells, on the 7th day, on the 10th and 14th, 3 doses increased 3 times the median survival. Moreover, with treated cells, even with the alone trastuzumab, which is not commercial antibody used for the treatment of ovarian cancers in human, and the median survival was 100 days. So our radiopharmaceutical was much better even than commercially used antibody now. But when we combined this immunotherapy together with our radiopharmaceutical, the median survival was even much better. So that's why now, together with Belgium and their company, they are focusing on the actinium-225. However, the problem is access to actinium now. And what else what we do? Despite those prosthetic groups that I already mentioned, we now are working on the bamboozerials, which are compounds which can chelate iodide. Iodine anion. Normally, iodide is introduced into biomolecules by nucleophilic or electrophilic substitution of benzene ring. Here we have chelator, so we want to check it if it's stable on in vivo condition, and attach also to biomolecule. We work on the nanomicelles of converting nanoparticles. All of them are containing radioisotopes. So this is for the multimodal diagnostic or therapeutic form. In NCBJ, we have also flash therapy based on the in-home build-up accelerator. It emits gammas or electrons, and compared to the commercial accelerators, they emit those gammas and electrons at very low rate, which means to deliver a high dose, it requires like half an hour, 1 hour. Here, the dose rate is so high that the same dose can be delivered in nanoseconds or milliseconds. And the current studies shows that this flash therapy is very promising because it kills cancer cells but spares the healthy cells around. So we are working on this topic, and we would like also to combine with the Jagiellonian University and Institute of Nuclear Physics here in Krakow for the proton flash therapy. And we have also collaboration with Institute of Nuclear Chemistry and Technology in Warsaw and Technical University in Białystok for the application of various hydroxycinnamic acids, either for the therapeutic purposes for cancer cells, but also as scavengers of reactive oxygen species, which are formed in the, let's say, our skin after exposure to UV light. And we have a very current situation with the equipment. We have very good infrastructure. We have our own radiochemical laboratories with permission from National Atomic Agency to work with more than 100 types of radionuclides. We have very well-equipped biological laboratories and also the SPECT camera, and soon in March will be delivered PET-CT camera. So if someone is interested in some animal studies, we are very open for the collaboration. And thank you very much.
So we have a time for question for discussion.
I have a general question about
yes.
So is radiopharmaceuticals. So the element will radiate since it's produced, right? And it continues radiating until it decays, and it's a normal non-radiative atom. So since you synthesize it, how do you deliver it to the patient without losing it on the way?
That's a very good question. So, for example, that's why some of the radiopharmaceuticals can be produced in the hospital. That's why many hospitals, for example, currently have cyclotrons by their own, the small ones, so they can produce fluorine, gallium, zirconium. And at that moment, they just extract the radioisotope and try to combine with the carrier, with the molecule. But in this situation, it's only for the simple radiopharmaceuticals because the hospital cannot make so many manipulations and use the automatic synthesizers, for example. In case of those radiopharmaceuticals, which are, let's say, produced centralized and later distributed, there are several ways. If the half-life of the radionuclide, it's long enough, like 7 days, it doesn't matter. It's just the production in few hours, dividing for the special packages into hospitals, and they are shipped. Of course, we assume that the radioisotope decays, so that's why the higher dose is prepared and shipped to the hospital. So whenever the package will arrive, whatever decays, still there will be enough radiopharmaceutical for injection to the patient.
Okay.
But still, we have to keep in mind that when it decays, the radiolysis is occurring. So there are many protectants also added to protect from the radiolysis effect.
Right.
Mhm.
And also, once it is, and it's a neutral or like non-radioactive atom, it will be filtered to the kidneys, or it can stay in the human body just as a neutral element.
Mhm. I mean, even if it stays in the kidney, it's not stays like forever. There is some washout. Either it's faster or it's slower, but at some moment it will be eliminated from the kidney. As in case of small molecules, the big molecules accumulates in the liver because this is the way to remove it, hepatobiliary way. But still, they are metabolized. It's the matter how fast is it. We call it biological half-life. Mhm.
Okay, many thanks. Well, I have a question about this part, which can be modeled somehow here, because, it's. I think that what may be interesting is just to make a kind of simulation how this radiation is propagating in this part of the human body. Yes, it could help somehow to take decision where to put it and what type of this radionuclide should be, let's say, implemented in a kind of situation.
For modeling, there are, I think, several points where is the option for the collaboration. I'm just looking for a slide. Yes. For example, here, we cannot avoid animal studies because every molecule is totally different. So we need to, of course, we can do the imaging, but at some point we also have to know each time point what's the activity in the organ to calculate the dose. If it's below the threshold estimated based on the external radiation, or it's above. So this is for modeling. That might be one of the options. Shall we divide this single dose into 3 smaller doses? That might be a question for modeling. And the other for the modeling groups, we have several prosthetic groups. And prosthetic groups are those compounds. SGMIB and MAGIC are prosthetic groups. We just try to find a way how to stably attach the radioisotope, but also somehow modify properties, for example, to increase or to slow down the pharmacokinetic. But to synthesize them, it takes a lot of time. So for modeling, it might be helpful, like we can propose several structures, and the modeling can say, okay, maybe this structure will be better based on some databases already available. And so pharmacokinetic will be faster. Synthesis shouldn't be difficult. Incorporation maybe also. So there are many other factors which might be included. Moreover, if we synthesize this, we need to later on attach to the biomolecule. And when we attach, we don't know if we modify somehow the structure of the protein. So that also, now we have the discussion with one of the group from Warsaw University. We don't want to do the modeling on the big molecule because it's too much complicated, but on the peptide, which is a 6 or 7 amino acid. And peptide is quite specific because it has cycle form. Because between there are 2 cysteines which form the disulfide bridge. So we developed a special prosthetic group which cuts the disulfide bridge, and this prosthetic group substituted. So the ring can be either bigger or a little bit smaller. The question is how this prosthetic group will interact with the entire peptide if it's still the biological interaction with the receptor will be kept or it will be somehow destroyed. We can do this experimentally, but it's too much time. Maybe modeling group, because we have several prosthetic groups in our mind, longer, shorter. Maybe modeling can support us just by introducing, peptide interaction with the receptor is already known. It's in the protein data bank. But if we add some small fragments, how it will interfere with the structure and the energy of this peptide?
Oh, yes.
I cannot answer. I'm chemist, but I'm not familiar with the modeling methods. But maybe this someone from you know someone, or one of you is able to do it somehow, calculating this modeling if the structure is still kept in the same form, so our peptide still can interact with the receptor SSTR2. Despite synthesis of 50 different prosthetic groups, maybe we can synthesize
Yes,
only 5 of them.
Mm-hmm. Oh, yes, so it's...
Mhm.
So you mentioned that you do animal experiments.
Mhm.
Do you analyze the data from the animals, or do you catch the issues that you then go and see what was the biology change? So once you apply the treatment,
No, probably you ask if we do any Western blottings or something like that.
Yeah.
Later, those data we can use for any omics.
no, I'm just checking, because if you want to go back and understand how your performance are going to work, right, you need to see what was the biological response there. Like,
Mhm.
so you do the experiment, you run the experiment. I would say it's similar with what we are doing on aging, for example. So we are using animal data from liver this morning, and then we are able to reconstruct how the biology was working when the treatment was applied. Here, in your case, Paul, we could understand what are the interactions that you have, how they are changing, and measure and quantify them. For example, using entropy of the model.
Okay, in our case, when we use the radiopharmaceuticals, let's say for the treatment,
Yeah.
we have more or less direct effect. We see the destroyed cells.
Yeah, but you destroy the cell, but at the same time, you don't quantify the biology. So you see that they are destroyed,
Mhm.
but you don't measure the impact, biological impact that you have around the cells, or how the biology.
No. No.
So that's what I'm saying. So if you don't look, so for example, you see the effect, you feel the cells, right? But at the same time, you were saying, yeah, that some of the things go on the liver, some of the things on the other part of the body, but you don't really quantify that. You just you feel it them. That's why that's a good answer. So that's what I'm saying. So instead of just only looking to that, if you look into the biological effect, you could also inform what you are trying to design initially.
Okay, I got the point, because first of all, if we see the destroyed of the tumor, it's good for us.
Yeah.
But we also do the slices of, let's say, kidneys, and we check if there is any toxicity or not. It's just eosin staining, and we can see by ourselves, or we can send it to the external company which is qualified and specialized in doing this, so they can just tell us, okay, it's a 4th grade of toxicity, or it's mild toxicity, so everything should be fine, let's say, with the upscaling to the human. But the other problem, which somehow was already noticed, that let's say we target the HER2 receptor on the cancer cells, but there are many other receptors like HER1, EGFR called HER2 we target, but HER3 and HER4. So if we target HER2 and we try to destroy the cancer cells, somehow the other cells stop production of the HER2 overexpression, and they use HER3 for, again, giving the same signal to proliferate.
It's called homeostasis. So
Exactly.
that's the sign of information. So you have information, and then you have pick, and then obviously you can pick a place, and you get a balanced structure. But in reality, the person is not healthier. Just a balanced structure that you get there.
But I think this will be like a next step. I mean, we are just so far a 7 members group, and we do the initial studies now. But I think we shall collaborate with other groups to get more information. Definitely yes, because that's why the resistance of the cancers appears.
Yeah. And that's why you have the processes and so on. So
exactly.
The things, if the liver answers in a certain way, you have things. I think the thing that looking just to a single signal, it's a quick way to have an answer, but at the same time, it's also a quick way to not understand what you're doing.
I mean, it's a 0-1.
And biology is 0-1. They're also grayscale.
Yeah. But for this, indeed, it requires human resources, funds, infrastructure, and instead of doing this in, let's say, our institution, it's better to go outside for the support and collaboration. However, another problem might be we're working with the radioactive material. So sometimes we want to collaborate, but let's say we have organs and we want to ship it somewhere to another institute, but they say we cannot work with the radioactive material. You have to wait until it decays. So that's why we freeze them, for example, in the liquid nitrogen and keep it until decaying. But sometimes it doesn't help. So, yeah, sometimes we cannot continue some other studies because of that.
And in the case when it is application is for medicine, what about the certification process? So it requires some, let's say, some experiments at clinics. All this stuff should go on with some, let's say, group of patients and something like this.
Yes. So before getting the certificate or approval, it goes many clinical trials, phase 1, phase 1 plus, 2, 3, even 4. With every phase, the number of patients increase. It's mostly we don't do this as us because we are scientists. These are doing the companies. Which, let's say, something is developed, either they purchase the license and go further, or because it's too costly. But there is, I have a friend at this Warsaw company, and for example, they develop a new diagnostic radiopharmaceutical for the heart checking. It's a long process. It's a long process with every promising result, they have to approve for another clinical trials, get the approval, find the money, and so on and so on. So
it's outside of.
In most cases, it's outside, unless there are some companies which are, let's say, waiting for that some patents will. And they try to find a way to produce something similar, but in a more generic way. So at that moment, maybe it's easier.
But what about the situation when you have the same radioactive material, but let's say put in different chemical, let's say, surrounding, in the sense that it could be like on this figure, you will have something which was in one of these compounds, and you just put the same into another compound, which you expect that it will, for example, with binding, will be more efficient.
But you mean radionuclide or the same prosthetic radionuclide?
The same radionuclide, but different, let's say, environment for the radionuclides. So from the point of radionuclide, it should be the same procedure.
For a radionuclide, I mean, it doesn't matter if it's in the cells or it's in the water or it's in the tissue. I would say it's the case. So the physics is the same.
Yes.
Of course, maybe the density of material may influence on the distance of the emitted particles. But still, physics is the same. The chemical, the biological response is definitely different. Yeah, I mean, for example, why we focus now on the alpha particle radionuclides? Because beta minus are very good, but for the big solid tumors, like 2 centimeters, 1 centimeter. So if it accumulates over there, and let's assume there is also blood vessels inside, so we can deliver the radioisotope with the radiopharmaceutical over there. So it should be homogeneously distributed. But if the metastases are appearing, so they're small, we cannot apply the lutetium, which has, or yttrium, which has like 2 centimeter range of beta minus, particles. Because at that moment, the majority of energy is outside of the cancer metastasis. That's why alpha particles are more promising, short range. So, of course, there are many other factors like hypoxia and normoxia. If there is hypoxia, radiopharmaceuticals are working in both ways. First, release of the particle which destroy, but also during the release decay, there is ionization around. Whenever there is ionization, if there is oxygen, so reactive oxygen species are formed. They are also toxic. They can interact with the cells and can destroy them. If there is normoxia, which means that oxygen is homogeneously distributed in the cancer, this process occurs homogeneously. But if we have normoxia outside of the tumor and inside hypoxia, where is less oxygen, much less reactive oxygen species are formed, and this part of the tumor becomes not destroyed. So there are many factors. It seems on the slides that it's easily to just inject and distribute, but it's not always. I didn't present here, for example, because from those actinium studies, indeed we checked the distribution in tumor and in liver. In tumor, we had a very nice homogeneous distribution. In liver, there was very minor activity, but still it was homogeneously. In kidneys, we observed heterogeneous distribution. By organ, we saw that there is activity, quite a lot, but when we calculated the liver dose, it was much less before the threshold, which means we shouldn't cause the nephrotoxicity. But when we do the slices and put on the special camera, we saw that only on the cortex there was a high accumulation, and in medulla, most of the part of the kidney didn't have activity, which means locally dose was delivered probably much bigger than the threshold. By measuring just the organ, we didn't know where is it. So we could say, okay, the kidneys shouldn't be destroyed. But when we figure out only on the outer space, there was a lot of activity. So we knew that definitely we are in trouble, I would say. So yeah, that's why it's very necessary to do many techniques.
Very small question. Maybe this is a little bit out of scope, but is there any way to create the alpha particle once it's inside the patient with the laser beams that you were talking about, the electron beams?
No.
Or activate it in some way just with the laser? or heat it up or something? That would be the last part that you said with the electron beams.
For the radioisotopes,
No.
it's impossible because when you, let's say, put that target material together with the biomolecule inside, and you start to irradiate externally with protons. Protons also have to pass through the tissues, so they will lose the energy. And to form the radioisotope, there are some cross sections in reaction, which means at some certain energy it is produced. We can cause some more troubles by irradiating with protons than production of the radiopharmaceutic. However, this is very good point because there is a boron neutron capture therapy, where it was, I think, in the 70s postulated that boron compounds are delivered, for example, to the brain tumors, irradiated with the neutrons. Neutrons are, let's say, neutral for us, but whenever the neutron will enter the boron 10 nuclei, it forms a very unstable boron 11, which decays to lithium and alpha particle is emitted. But at that moment in 70s, there was a huge problem how to have a focused neutron beam. Because with the protons and electrons, you can focus the beam by the magnets, electromagnets, because they are charged. With the neutrons, they are neutral, so you cannot focus this. So now there are specially developed accelerators where you irradiate with the proton some material, proton is absorbed, neutron is released, and it's released in the form of beam. So that's why now the DNCT therapy is, the idea is renewed.
Right.
Mm-hmm. So this is a very good question. Of course, not for the irradiations with protons, but for the neutrons, yes. That's another option to form the alpha particle inside of the human body.
Okay. Well, I think that there is still time for discussion after the seminar will be
With
over.
coffee. So thank you very much. I would say that before, when I arrived, I had many discussions with some of you, and for me it was like a magic. I assume that this topic is somehow also some kind of magic for you, but yeah, I hope that there will be some potential collaboration, or if you are interested in some topics and want to know more, just feel free to ask me.
Okay, many thanks. Well, thank you. I would like also to thank our online participants. And the next seminar will be in 2 weeks from now. So, because in a week from now, we will have our internal meeting. So many thanks.
Thank you.